Hello everyone and welcome to the latest MDPI webinar on cells, which is entitled Nervous System Development and Plasticity in Model Organisms. Thank you very much for joining us today. This is the third webinar of a series that is based on special issues and sections of our Cell journal. So if you wish to submit your paper to any special issues related to this webinar, please check the webinar's website. Our chair today is Dr. Sepand Rastegar from the Karlsruhe Institute of Technology in Germany, and we will have two speakers. Dr. Ciaran Murphy-Royal from Université of Montréal in Canada, and Dr. Laure Bally-Cuif from the Institut Pasteur in France. At the end of the session, and actually after each speaker presentation, and then at the end of the session, we will have a Q&A session during which we'll address questions submitted by live online audience. Now I'd like to introduce our chair very briefly. So Dr. Sepand Rastegar is currently working at the Karlsruhe Institute of Technology, and his research focuses on the molecular mechanisms that regulate zebrafish neurogenesis and regeneration. His models are the spinal cord and the adult telencephalon. Now I hand over the floor to our chair, Dr. Sepand Rastegar, and to all of you, enjoy.
Okay. Hi, everybody. It's not easy to talk from the screen. I'm not really used to that. I will be very short. I'm very happy to host together with Cell this webinar, and I thanks everybody who joined that. I want also to thank Yvonne Fang, who actually with whom we organized, and she helped me the first step of this webinar. I think it's important to thank the people who help. I will just briefly to introduce the two speakers. I first also thanks Laure Bally-Cuif, who accepted to give the talk. I also Ciaran, both of them, for accepting to give the participant to give a talk here. Very briefly. So I will. Laure will talk at the second part, so I will introduce Laure later. I start with Ciaran. Sorry, it's difficult to do that over the screen. I'm not used to that. Ciaran Murphy-Royal, who is actually Irish, and he studied in Ireland. After his first study in Ireland, he moved to France in Bordeaux, where is really one of the French hub of neuroscience, where he started to work with astrocyte, and then from there, Ciaran, you can correct me if I'm making mistakes. Then he moved to Canada in Calgary. He went on to work with the astrocyte, where he investigated the effect of stress on astrocyte. And now since last year, I think he has his own lab in Montreal. We have some common point with Ciaran is that he's speaking French, although he's Irish. So this is one of the link. And I also repeat myself, but I'm very happy to have him because I listened to one of his talk and he gave a very nice talk, nice data, and I'm looking forward to hear him giving now this talk, and thanks for accepting my invitation. Please, Ciaran.
Well, thanks, Sepand. That's really kind words. Yeah, I'm really happy to present my work here. It'll be, I guess, similar to what you might have seen before, Sepand. I think the lab we've been open since June, so starting right in the middle of the pandemic. That was really great. But yeah, so I'm going to show you the data which fits into the plasticity element of this seminar series. So yeah, so basically today what I'm going to present to you is the work that I just recently published in 2020, looking at how astrocyte network dysfunction regulates how synapses respond to stress. So kind of linking the themes of astrocytes, synaptic function, and how these can be modulated by physiological conditions. And so yeah, it's really happy this fits into the whole scheme, I guess, of nervous system development and plasticity. So I'm more of the plasticity in this case. To start, yeah, so I just want to start with the acknowledgements from this work. This work here has been, I guess it was carried out in mainly J.D. Baines, Grant Gordon's lab at University of Calgary. We had nice collaboration with people in the States and other places, other labs in Calgary. So this is kind of just some nice pictures of the team, kind of reminiscing to the nice big mountains that we had in Calgary, which are not as big here in Montreal. But anyway, so what my lab is looking to study, and what I kind of continue to do now, is looking to the central adaptation to experience. So how experiences are perceived by the brain, stored, and how this induces a response. And so when we think about the central adaptation to experience, so if you think about the brain, we can think about the brain as a network of networks. And here you can see, in general, you get an experience or event that's perceived by the brain, of course, comes in through sensory organs. And we get changes in synaptic activity. So you might get some brain regions becoming more active, others being less active. And in general, this leads to some sort of adaptation. And so I'm not studying this phenomenon on kind of a brain-wide scale. I'm kind of picking brain regions I'm interested in and going from there. And so I'm looking at the synaptic response to stress. So when you get an experience coming in, you get a presynaptic release of neurotransmitter, activating a postsynaptic response. And if this is in terms of a learning and memory experience, you can get plasticity of these neuronal circuits. So we can get depression, so we can get a reduction in the strength of this synaptic connection, or we can get an enhancement, which could be due to presynaptic release, postsynaptic insertion of receptors, or we can get this bidirectional change in the strengthening of these synapses. So that's the basis of the synaptic plasticity forms that I'm studying. And so when we're thinking about the brain, I just showed you pictures of synapses here. When we think about the brain, of course, your neurons is the first thing that comes to mind, but I don't have to tell you. I guess this is a kind of the topic of this is a cells seminar. So we're thinking about all different types of cells in the brain. And so the cells that I'm interested in really are the glial cells. So thinking about the astrocytes here is just a nice marker using GFAP, one of the cytoskeletal proteins. You can see how nice that cell looks. There's also microglia in the brain. They're more of the, I guess, classically assumed to be the resident immune cells, but have lots of basic vital functions, such as synaptic pruning during development. And there's also oligodendrocytes. So oligodendrocytes forming in the myelin. And I guess this is oligodendrocyte studies are really big in fish, which is what I know Laure has studies a lot, because of course fish are quite transparent until a certain age. So you can study glial cells in all different model organisms, not just mice like I do, but actually fish are really good for specific questions too. And so if you think about astrocytes in the central nervous system in mice, at least astrocytes and humans tile the brain. So here's like a rainbow image. So it's fluorescent promoters, fluorescent proteins under astrocyte promoter, and you see that they just kind of line the whole brain, a carpet of astrocytes everywhere. And this is the same throughout any brain region that you look. And if you look at a more small scale, you can see individual astrocytes here, some in red, some in green. They occupy their own territory or domain. They have an overlap with their next door neighbor. So this yellow section here is the overlap between the astrocytes, so about a 10% overlap. And they form a spherical volume in which they control all the synaptic activity, the blood flow, these kinds of things astrocytes are controlling in their little domain. And so astrocytes, when they kind of look like this, I mean, when you look down a deep level, they look more like a sponge. So these kind of sponge-like fine processes go in and they wrap around synapses, and you get this really nice morphology. This is kind of more representative of what an astrocyte really looks like compared to the star shape that's classically associated with these cells. And in this case, because there's such a sponge-like really fine processes, you can see here in this electron microscopy image a presynaptic cell in yellow, postsynaptic in blue, and we have the pink here is the astrocyte, which gets right into the synaptic cleft. And they're interacting with both pre and postsynaptic elements. They're releasing gliotransmitters, and they're taking up neurotransmitters, all these kinds of things that I've studied before. And this is how they do it, is by getting right in close and tight into the synaptic cleft. And so astrocytes, one of the important components is that they line the cerebral vasculature. So it's really important for controlling blood flow in the brain. And as you know, blood flow in the brain is quite important to fuel all these high energy demanding processes. So the brain is a highly vascularized organ. And I mean, as you might have heard before, for only 2% of the body weight, it uses 20% of the energy expenditure. And so it's a really highly glucose demanding organ. And so astrocytes, if you do a stain, this is a transgenic EGFP astrocyte mouse, you can see here that we're actually, the astrocyte end feet are lining the blood vessels. So you can see this arteriole here with the branches coming off, and the astrocyte end feet are wrapping around the blood vessels. They can release vasoactive molecules to cause dilation or constriction of blood vessels in response to nearby synaptic activity. So they're listening to synapses with one arm and controlling blood flow with the other. And so this is important for many different reasons, as I said. I mean, you can think about the really large blood vessels coming into the brain. There's only going to be a certain number of astrocytes grabbing these large blood vessels. And so to shuttle the energy that's coming in down these big pipes, astrocytes do this through their interconnected networks of cells. So astrocytes don't communicate like neurons in the kind of chemical synaptic transmission. They form gap junction channels, so pores between their next door neighbors into groups of about 50 to 100 astrocytes. And in this way, they could get energy from this blood vessel all the way to the synapses nearby that need it. So they can bring molecules to the synapses, and they can also take away waste at night. And that's kind of glio with Mike and Nadergaard's glymphatic system, where the astrocytes clean the brain at night. And so the metabolic role for astrocytes, as I mentioned, they have one arm on the blood vessel, one arm on the synapse. And so the link between these two, taking a quick look through what happens, you get glucose being taken up from the blood by astrocytes. Astrocytes are the only cells in the brain that can actually store energy, so they can store glycogen. And so no other cell actually has an energy reserve but astrocytes. And this glycogen can be broken down again through glycolysis, making ATP for astrocytes themselves. So they use this to maintain, I guess, to power their own glutamate uptake and other ATP-dependent processes. But the pyruvate that they make with this doesn't necessarily get turned into ATP. Rather, they have specific enzymes that convert pyruvate to lactate. And this lactate has been shown then to be shuttled through monocarboxylate transporters that's given to neurons. So there's some in vivo evidence showing that this actually does happen, that there's like a gradient of lactate from astrocytes to neurons. And once in the neuron, the lactate can be quickly converted to pyruvate, brought to the mitochondria to produce abundant amounts of ATP. This ATP is really important then for reversing or maintaining a hyperpolarized membrane potential, for powering all the sodium, potassium, ATPase pumps, all these things that neurons need to do to maintain proper synaptic transmission and plasticity. So the neurons are the main consumers, and astrocytes are just making sure that there's enough energy when needed. And of course, neurons can take up glucose themselves in the extracellular space. That's undoubtedly true. They can use this to make energy, but it seems actually what the glucose that neurons are taking up is usually made, is I guess predominantly used, or it seems to be, to make as an antioxidant. So basically making NADPH to reduce the reactive oxygen status of these cells. And when they're producing lots of energy, you need lots of antioxidants to kind of balance that out. And so one really nice study in 2011, one of the most convincing, showing that there is an astrocyte to neuron lactate transport. It's required for long-term memory. They looked at synaptic plasticity and memory. And so in this case, they blocked glycogen breakdown, they blocked the lactate transport out of astrocytes, and they blocked the lactate transport into neurons. And each one of these steps independently was able to block LTP, so synaptic plasticity, it's potentiation, and it also blocked learning and memory in vivo. So just kind of a good idea that this is very important for certain high energy demanding processes such as learning and memory. And so what we're thinking about now is this kind of process in our mind. It's really important. So we're thinking about these experiences or events that change the synapses and lead to adaptation. And so that's what I showed you before. But of course, now we're seeing that the astrocytes are involved in this mechanism somehow. And so we're getting, an experience or event going to neurons. We might get astrocytes listening to neural activity, inducing some changes, releasing something. Maybe it's lactate, maybe it's another gliotransmitter, and you get the adaptation. But coming from my background, where I've studied glial cells and specifically astrocytes for most of my career, I'm really interested in seeing if there's maybe some experiences or events that can change how astrocytes interact with neurons. And so something first comes to the astrocyte, these cells respond and manipulate neuronal output. So I'm wondering if there's something that could be responsible for that, leading to the adaptation. So kind of taking an astrocyte-centric approach to learning and experience in the brain. And so I kind of bring up this example every time because I'm in Université de Montréal, and so Hans Selye was a Hungarian neuroscientist here too, and he coined this stress and general adaptation syndromes in 1950 in the British Medical Journal. He stated that all organisms can respond to stress, and as such, it's the same basic reaction pattern irrespective of the agent used to produce the stress. So of course, that's kind of a broad, vague statement. But in the end, it's actually, when I was thinking about it, even preparing for this talk over the weekend, I was thinking about how my work could fit into, people who work more with maybe zebrafish. And the thing that I'm studying, of course, from the title, you could tell is stress. And of course, here we're talking about stress. If you look at the hormonal response to stress, so glucocorticoids, it's the same process, whether it's humans, mice, or fish. You get hormones produced in the periphery outside in a sort of adrenal gland. It's not exactly that in the fish, but it's a similar type of gland that produces hormones that get back into the brain. So this mechanism does really hold through, and it's robust throughout many different species of animals. So super interesting thing to study, and maybe super translatable between different animal models. And so when we're thinking about stressors, we can think about real stressors, predation, starvation, inflammation, dangers, or perceived stressors, hassles, deadlines, major loss. All of these tend to converge in certain brain structures. You think about the hypothalamus, the HPA axis, to induce this stress response. The stress response, and you get the hormones produced, feeding back into the brain, and they act throughout the whole brain to change synaptic and brain activity in general. And so when you think about this, just kind of a more structural image here, you see where psychological or physiological stress, you get convergence on specific brain regions to produce these stress hormones. And that's what I'm kind of trying to get at here. And the timeline of stress is a nice thing to study because you have two distinct time points where right after the stress, you get the internal hormonal response. So what's happening inside of the brain, changes in glutamate, GABA signaling, noradrenaline, all these CRH and other hormones changing inside the brain. And then at a more distal time point, you get the feedback mechanism, so the glucocorticoids, which tend to shut things back down. And that's occurring, let's say 30 to 60 minutes after the stress happens. You can kind of differentiate what's happening immediately versus the late effects. So it's kind of a nice phenomenon to study in that respect. And so stress impairs synaptic function. We know this, it's been shown many times, it's super robust finding. If you look at hippocampal brain slices, you record just synaptic activity here. You can see basal activity, you have high frequency stimulation. This causes the synapses to potentiate, to get stronger. In naive conditions, you see a nice potentiation. And in stress, I guess in this case, they got an animal, they gave it some foot shocks, then made the brain slices. And so the animals were stressed prior to making this brain slice preparation. You can see that the synaptic response to this stimulation is much lower. So you're getting like a dampened stress or synaptic response after stress. And this, so in that case, it was foot shock. You can do, and that was kind of taking the slice out of the brain. You can look at this in the brain in situ. You get a rat in this case, they put it on a platform. So a different type of stress, there was no pain associated. It was just left in an exposed environment. And you recorded in vivo LTP. And what they found in vivo was the exactly same phenomenon. So you get this reduction in the synaptic response to high frequency stimulation. So classical LTP following acute stress. So single bout stress, not even a chronic stress happens. And so then it comes to the question to me, I'm so interested in astrocytes and stress, could astrocytes be involved in this mechanism? And so if we think about the hormonal component, so here we're thinking about the glucocorticoid receptor. If you look at mRNA expression levels, I'm just showing astrocytes and neurons courses of the brain cells that have it too, but astrocytes express a lot more than neurons of this glucocorticoid receptor. Same goes for the mineralocorticoid receptor, which is maybe less involved in stress responses, but it's a glucocorticoid receptor, this low affinity receptor that binds the high levels of glucocorticoids that occur in stress. And this makes lots of sense to me, considering what I showed you earlier too. Astrocytes line all the blood vessels, and these glucocorticoids get into the brain through the blood. They're produced in the adrenal glands. And so one of the first cells in the brain they're going to come in contact with is likely an astrocyte. So to me, this all kind of starts to fit together that there could be a role for astrocytes in these synaptic phenomena. And so we know that astrocytes are stress sensitive. Think about stress and stress disorders. What we have is depression is one of the most commonly associated psychological disorders associated with stress. And in human brain tissue, there was control astrocytes that they stained. They looked at astrocytes from depressed suicide patients. And what they found is the cell body area was changed in size, got bigger, and also the total length, so the branches got bigger. So it's kind of an indication that these stress-associated disorders in humans, actually astrocytes are implicated on a clinical level. And so this brings me to the question, what I wanted to answer is how to define a role of astrocytes in this stress-induced impairment of synaptic function. So how do astrocytes, how are they affected? And how does that change synaptic activity? And so we started off by targeting astrocyte-specific changes. So a broad screen using RNA-seq. So it's not a single cell, it's more of a bulk. We took the somatosensory cortex in this case. We had a transgenic mouse line expressing a Cre recombinase under an astrocyte-specific promoter. We cross it with another mouse line, which has a hemagglutinin, a HA tag, which is specifically put on ribosomes. And in this way, we're able to pull out mRNA from astrocytes. So in our hands, we made the mice swim for 20 minutes. They really don't like that. That's a significant stressor for a mouse, which is an animal that doesn't usually swim in the wild. We waited 90 minutes, and the idea of that was seeing if the glucocorticoids were getting back into the brain. And then we pulled out ribosomes and sequenced ribosomes. So when we got the mRNA, we can take out the specific ones that are astrocyte specific. And you can see in this prep, we just showed that it is specific to these cells because you don't find candidate genes for other brain cells. And so when we looked at this transcriptome, we found that only just over 100 genes were changed. So it wasn't a crazy, huge change after a single bout of stress, which makes a lot of sense. And looking into this list, this is the entire list of everything that changed. Not too many things jumped out to me from my perspective, from my bias of the things that I like to look at. I was kind of a bit disappointed when I saw this data. It didn't seem to kind of fit with what I thought was going to happen. But we looked into this from canonical signaling pathways, so grouping these genes into their signaling pathways to see what would change. And some interesting things actually did pop out. So the top hit, so the highest changed signaling pathway was the Wnt beta-catenin signaling, which is really interesting. Of course, lots of people know that for development is really important. And other mechanisms that were, I guess, commonly associated with cancer signaling. So to me, that doesn't necessarily suggest we're inducing a cancer, but maybe more cell morphology changes, which is what I was just showing you in the human brain tissue. So potentially indicating that there. So we followed up looking at the structural plasticity. We can make a mouse swim. We made some brain slices, and I patch clamped an astrocyte and filled it with dye. So in this way, I'm able to target a single astrocyte. I can fill it with a green dye and get really nice single cell morphology. And so you can see here we have naive on top. Yeah, I use the red dye SR101 just to label astrocytes, just to target them. Once I fill them with the dye, then you can see we're able to fill up all the arborization, all the branching of the cell. We can look at the territory size. We can also look at ramification indexes. And what I found was that after the single acute stress, we saw about a 10% increase in the size of the cells. They got bigger. And we also saw they became more branched. So kind of a good indication. If structure changes, maybe function is changing too. And so we next looked at this Wnt beta-catenin signaling pathway, the highest hit, what's going on there. So we found a number of genes that were interesting. And what was interesting to me, what stood out in this signaling pathway is that it can control connexin 43, which is one of the major gap junction channels in astrocytes. So this allows them to form those networks, those metabolic networks which shuttle things between astrocytes. And so we looked at we did Western blots for different astrocytes, connexin gap junction protein channels. We looked at connexin 30, connexin 43, they're specific to astrocytes. And what we found was that we saw a significant decrease in connexin 30, but we didn't see a significant decrease in connexin 43. So it was just kind of a basic overall kind of idea what's going on. Could there be functional changes? And this was from bulk brain tissue. So we then thought, okay, maybe it's what's in the membrane that counts. It's this little kind of, I guess, pocket of connexin 30 and 43. And in that paper that we published, it was the same finding. We looked at the membrane fractions. So it seems that we're not too heavily influencing the protein expression levels, but still there are many different ways to affect function in these processes. And so I wanted to look at how are they working? Could this small change in the levels of protein changes result in large functional changes? So to look at gap junction coupling to astrocytes, I mean, you can see here just the example again, just a couple of astrocytes labeled with a transgenic dye. They do connect to their next door neighbors, have this overlapping domain. Classically, if you patch a single astrocyte and let it fill with biocytin, you can label lots of different astrocytes, count the number, the whole size of the territories, and this can be used really nicely to define the size of astrocyte networks. But I wanted to do it in more of maybe a functional or real-time readout, and so this is what we did with 2-photon imaging and patch clamp microscopy. I did the same as before. I used an astrocyte marker. I'm going to patch an astrocyte, and I'm going to let it dye, a small molecular weight dye that passes through the gap junction channels to fill the neighboring astrocytes. And so you'll see that here on the right-hand side, we're using Alexa 488. This is 5 minutes sped up into a couple of seconds. You can see here, I patch the astrocyte. Initial one, you see we start to load, you see the start to branches start to show, and all of a sudden you can also start seeing the next door neighbor astrocyte starting to fill with the Alexa dye too. So with this approach, we can look at the coupling in astrocytes in real time, and not maybe just getting an idea of how many cells are in the network, but more how rapidly they're communicating with their neighbors. So to quantify this, it's really kind of straightforward. We take regions of interest and look at fluorescence intensity changes over time. At the patch cell, you can see the fluorescence intensity goes up, remains stable, so the dye fills the cell body. If you look at the neighboring cells, so the dye flow, you can see that the fluorescence intensity kind of increases slowly over time in this. And so we can quantify this and create a tau curve, so a fill time, using a mono exponential on that curve. You can see here, just this is the black is the average, gray is the raw data for maybe 30 different patched astrocytes and even more neighboring astrocytes. So the gray is the actual raw data. So you can see it's a really robust, reliable way to look at coupling between these cells. And so does stress affect coupling between astrocytes? We carried out the same prep as before, made them swim, made brain slices, start to patch. And in this case, I'm going to look at the neighboring cell, not actually the astrocyte that I've patched with the dye. And so what we see here in naive, you see immediately there's no dye in the cell, but it starts to fill up from its neighbor. And in the stress, in this representative image, you can see that the dye fills up a lot more slowly. And so we quantified this. We saw that there was maybe a 60% decrease in the coupling time. So a really significant decrease in coupling between astrocytes. So how does this occur? Again, you can look at the early mechanisms or the late mechanisms of astrocyte function. So if we take a naive astrocyte, we patch it, we look at the average fill time, you can see it's there. I took naive brain slices, I put them into corticosterone, the stress hormone. We saw an actual replication of our in vivo stress findings, so it seems to be glucocorticoids through that alone. If we used RU486, so it's the glucocorticoid receptor blocker, so specifically for that one, put cort on, we have no effect. But if we stress the animal and make brain slices immediately after the stress, so not letting the stress hormones be produced and come back to the brain, again, we get no effect. So it's not the immediate changes that's happening in the brain that's doing this. It is the hormonal feedback that is affecting astrocyte function. And again, the last kind of control group was using metirapone, so this blocks steroid synthesis in the adrenal glands. So they drank it in the water for 24 hours. I stressed them out. I let them wait 90 minutes. So hormones should be getting in, or even anything else that might be occurring in the brain over that long time span, and we saw no effect. So you do need glucocorticoids to get into the brain and act on glucocorticoid receptors. And in naive, that steroid synthesis inhibitor did nothing. So you can see here just the average fill curves from these groups. The right shifted one, meaning slower coupling, was the glucocorticoid treatment alone. And so does this affect energy substrate shuttling? Well, we can't look at D-glucose going through the network because it's not fluorescent, but there are fluorescent analogs. And so again, just to briefly go through this, I know I'm going to be cutting it short on time. This fluorescent glucose molecule showed exactly the same thing as what we saw with Alexa. It was right shifted, slower shuttling of glucose between the network after stress. And so we need to now that we have this phenotype that we show that stress does affect astrocyte function, how can we link this up to synaptic activity? And so to do this, I wanted to record synaptic activity through the astrocyte. So just some validation steps that you can do this. I'm patching an astrocyte under 2-photon here. I have a classic extracellular field electrode. Anyone who knows physiology, just measuring both synaptic activity, and I'm stimulating nearby. And so with this approach, you get the astrocyte response measured through the astrocyte, you get the classical response. And what I found by increasing the stimulation intensity, that you can get a linear relationship between these two. So through an astrocyte, you're measuring changes in ions in the extracellular space. So you're actually indirectly measuring synaptic function. And so we get a nice linear correlation. This was blocked by DNQX, TTX. So it is neuronal based. It's not an astrocyte synapse thing. It's a neuronal thing just measured through this cell. And this gives us really nice ways to manipulate astrocytes and then see what happens to the synaptic function. So one of our manipulations involves looking at the effect of transgenic modification of connexin 43. So we made this dominant negative, which would be overexpressed. It has a GFP on the N terminus to stop its insertion into the groups of gap junction channels. It also has a single point mutation blocking the pore. So it'll be expressed, it'll get into the groups of connexin 43, but it won't be functional. And so we did this viral approach. We injected the virus or a control virus. I looked at coupling between astrocytes in cells that expressed this modified connexin 43. And you can see after 5 minutes, these neighboring cells have no green in them. So there's no coupling at all. It completely blocks the coupling between neighboring astrocytes. So a really nice tool. We decided then to look at what happens to synapses. And in this condition where we block coupling, we see no synaptic plasticity at all. So this is a common, in the gray bar, I'm giving a high frequency stimulation. We should see synaptic potentiation. We don't. This is in the cortex. And so using this approach, I was able to put 2 millimolar lactate into the astrocytes, so give astrocytes the energy, normally get the energy from their neighboring cells, but now I'm giving it through the patch pipette, and we're able to completely rescue the effect on synaptic function. So it seems that with an astrocyte network dysfunction, which I saw occurs in stress, this kind of artificial way shows a similar phenotype, no LTP, because astrocytes are not giving the energy they need. And so does stress induce this energy deficit? We use lactate sensors. Lactate is what astrocytes release, I told you early on. We looked at in naive, you give a high frequency stimulation to neurons, lactate is super stable, you get nice homeostasis. In stress conditions, you get a breakdown of homeostasis. So it seems that there is a decrease in lactate. And we looked at different time points in the basal, there was no difference in lactate. Immediately after the high frequency stimulation, both naive and stress conditions did release lactate, but it was actually the sustained release was important for this synaptic plasticity. So its sustained release is decreased. So kind of seeing that lactate release is not just a one-off thing, it kind of needs to be sustained over time. And so now what happens to stress and synaptic plasticity in my hands? Of course, just showing you what I showed you earlier on, in naive conditions, I give a high frequency stimulation in the cortex, we get nice LTP. In stress, we no longer get LTP. That's been shown before, it's quite nice. So now can we link this back up to astrocyte function? And of course, we wanted to ask if lactate can rescue these effects. So we have stress, now we give that astrocyte from a stress mouse some lactate, and we can rescue this phenotype. You can see here just the effect on plasticity in naive stress plus lactate. So it seems that stress directly affects astrocytes through the glucocorticoid mechanism and impairs the synaptic response. And so, yeah, just the final word, that lactate needs to be taken up. It's not just that I'm doing something to astrocytes, it's actually being released and given to neurons. Because if I block neuronal uptake, so there's 2 drugs I used, sorry here, the ARC 155858 in red, which is a really neuronal specific one, and 4-CIN, and so it blocks neuronal uptake of lactate, and I don't get this rescue phenotype anymore. So it seems that the lactate I'm giving to astrocytes needs to be given away. And so the same goes for hippocampus. So of course, lots of the synaptic plasticity fans look at hippocampus. I did all the same measurements of hippocampus, found exactly the same things, that stress reduced coupling, reduced LTP, and I could rescue it. So both in the cortex and hippocampus, this holds true. And so just to summarize quickly, we have in general naive conditions, glucose is taken up by astrocytes. This glucose then can be shared between the network in the form of lactate or pyruvate between cells. If we get synaptic activity over in these synapses, you're going to get shuttling of glucose to this hotspot. If you get activity down low, it's going to happen over there. And so what I showed in this paper is that in stress, you get glucocorticoids coming in through the blood. This decreases the gap junction coupling between astrocytes. This impairs the flow of energy between these cells, so in the timely manner, in this kind of rapid necessary time that we need. And this leads to a synaptic energy deficit, so which is measured by LTP impairment. So to me, this is kind of a first instance of how this experience-dependent astrocyte plasticity. So stress is inducing a plasticity in astrocytes, so reducing the network function, which regulates the synapses nearby. And so yeah, so kind of the way I frame this as kind of a more of a kind of funny kind of way is that the neurons, of course, everyone's recording these because in the Wizard of Oz course, you get all the fire and bang and it's really nice and easy to see. And from my work, what I'm trying to show is pull back the curtain and you see it's the astrocyte that's behind the curtain doing all the things, making sure there's enough energy, making sure that everything functions correctly. So with stress, potentially astrocytes is where we should be looking for recovering the effects of stress, even in stress disorders. And that's kind of what I'm going to do with my work. So the future directions, I briefly say it in 2 slides, looking at these different diseases and disorders and modeling them in model organisms to see how we can look at stress and maybe relieve the effects. And so of course, all these things induce stress, all these diseases, but conversely, stress makes all of these things worse. So it is one of these vicious circles where stress, in addition to being itself and the result of itself, is the cause of itself. And so, yeah, we're looking at the influence of stress on behavior, how 1 stressful event, 2, 3, and kind of how this becomes, when this becomes pathological, repeated stressors, and we don't know what the limit is, the threshold. I guess it's different between people, different between mice, and if maybe different disorders could reduce this threshold from physiology to pathology. So in the lab, the first recruit was a great master's student. She's still here at Ifalua Adedipe, and she's looking at contextual fear conditioning in this case, looking at how animals learn over time, looking at the effects of stress on this. And I mean, with just this learning and memory paradigm, because it's a stressful memory, actually these memories are enhanced. So stress memories subsequent to a swim stress are enhanced. So it's kind of an interesting finding. And we see similar things to a chronic stress. So kind of really nice work that we're going to follow up on to see how all this synaptic work I just showed you fits into an in vivo model in a wake behaving animals. And so we're going to bring online as well some fluorescent lactate sensors we just developed. Actually, this preprint came out on this weekend, I think it was, where we'll have this new fluorescent lactate sensor, like a GCAMP style GFP for lactate. So it's quite nice. And so with that, yeah, I'd just like to finish on this slide. So thanks everybody for your attention. Thanks for the funding that we've got so far, and really thanks for the invitation. It's really a pleasure to present this work today.
Okay, so I think I'm the one to now take question. Thank you very much, Ciaran. Very, very interesting talk. I think I will be polite and leave people ask first question. I just check, I think. So there is first question from Amber, a friend that I know.
I'm quite interested in gap junctions in the glial cells, and mainly because there's a lot of evidence showing that these in seizure models in mouse, there's strong correlation between reduced gap junctions and seizures, also in human case of temporal lobe epilepsy. I know that you're not interested in epilepsy, but I would like to ask you still, are there any evidence for increased seizure susceptibility in any mouse models that are stress mouse models?
Yeah, so I've been looking really into some early life stress models, and there's lots of links between early life stress and seizures. So there's been, I mean, kind of surprising to me, but with kind of more developmental stress during development, let's say, you know, maternal separation, or these are limited bedding, kind of abuse and neglect style environments. But there is definitely a link with epilepsy and that, and kind of more higher susceptibility to seizures in these mice throughout the rest of their life. And I was even thinking about this question. It's funny you mentioned that. I was thinking about how, the kind of how gap junction channels, you know, decreasing energy, but you can get more activity through epilepsy. And so one of the reasons why that might be the case is that lactate's necessary for maintaining a low membrane potential. So you need lactate to, you know, when you have action potentials, you need to reverse. You need sodium, potassium ATPase pumps to bring the membrane potential back down to normal. And so lactate's really important for that. So potentially, if you don't have enough lactate or gap junction channel function's not working, you might see a hyperpolarization and a lower threshold for epilepsy.
Amazing, amazing, wonderful. Thank you so much.
Thank you.
Okay, next question. So maybe I ask this one. The question is from Feng Ru Tang, and he asks, can stress induce astrocyte morphological and number changes? Any good markers for such analysis?
Markers for analysis of this?
Yeah, sorry, no, go ahead.
Yeah, so I guess what I was using, I guess, is quite maybe not accessible to 2-photon imaging and the patch clamp. So other things you could use, yeah, there's lots of, you could do, I mean, viral injections of GFP into astrocytes, or even just do GFAP staining. You kind of see these effects with GFAP labeling in astrocytes. So patching and filling astrocytes gives you the nicest morphology, but even with these chronic stress models, GFAP, that cytoskeletal protein, you start to see changes already. You start to see, you know, denser processes and things like this. So even just that alone is a good indication that you're having some effects on astrocytes.
Okay, and then his first question, I mean, the beginning of his question was, is actually stress changing the number of astrocytes and their morphology or not, if there is any change?
Not in my hands, not with this acute stress. All the effects I saw quite nicely and reassuringly is that after even a single stressful event, everything went back to normal 24 hours later. So it's like a transient effect. But with the chronic stress models, you don't see this transient effect, more permanent. But yeah, for the acute stress, there was no change in astrocyte number, luckily.
Okay. Is there any other questions? Hot questions? I mean, we have still, yes, I see something here. So Judith, hold on. You ask the question yourself. It's too long. So Judith from Netherlands, can you, Judith, speak up?
Yes, beautiful work. So I really enjoyed your talk.
Thank you.
I was wondering, are there, because you mentioned that these astrocytes cover different, they are sort of tiled over the brain, right? So I was wondering, is there a difference in the actual area that one astrocyte covers over the brain? And a related question that I was just wondering about is within one region that one astrocyte covers, does activity of neurons within that field also modify the response to activity of other neurons within the same area covered by one astrocyte? Do you have any idea about these?
Yeah, yes, that's a great question. Yeah, so for the astrocyte sizes in different brain regions, they are all kind of slightly different, I guess. I can't say for sure that there's one size of all astrocytes throughout the brain. So I think there's definitely regional heterogeneity in the kind of proteins they express. So I'd imagine there is some heterogeneity in the sizes as well, and kind of depending on brain layer and brain region they're in. And then for your second part of the question was activity changes in size.
It was more, I was just wondering whether like neurons within the same area covered by one astrocyte, whether the activity also modifies the response of that same astrocyte to other.
I think it's not. I think there's not much being done on like independent synapses in the same domain. But yeah, in some instances, astrocyte calcium, let's say the calcium events showing that they're active, are quite local to where the synapse is. And then you might have some events where the whole astrocyte lights up. So you can imagine there's some cases where astrocytes are keeping the synapses independently functioning and completely separated, that might happen, that they want to, like, I guess, scale the synaptic activity in the whole astrocyte domain. So I imagine that it is possible. I think it's a really interesting idea, and I think it'd be cool to look at that for sure.
Okay, thank you, Judith. So I think I ask one question and then we stop, and then if I don't know, I think you have no problem, Ciaran, if people write you, if they have a very hot question and write you and ask the question themselves. You put your Twitter account there so they can directly contact you.
Sounds great.
My question is, if I understood well, so in this transcriptomic study that you show, it's basically you stress the animal and then you look what is going up and what is going down. Is it exact?
That's right. Yeah.
Yeah. Okay. I mean, it was very, very interesting because actually, you know, we work with the zebrafish adult brain and we do injury in the brain. And basically what I've seen is that it's the same. It's basically very, very close. I mean, we see the same first pathway to be activated is Wnt. Many of the genes that you show, it's very, very, so it seems to be a very similar things going on. Yeah. And my question, do you see also inflammation popping up? Have you looked on that?
I think it'll be something that I want to get into more with maybe the chronic stress paradigms. Definitely, I want to see if there's maybe an inflammatory component, as you said, like with these chronic stress. I mean, that is already kind of a field in itself, you know, stress, inflammation, astrocytes. But yeah, seeing if the chronic stressors are inducing more of an inflammatory phenotype is something I want to really look into. I mean, I think it is conserved too, right? I mean, between different animal models, even astrocytes in zebrafish. There's one of the best papers in the last few years has been on astrocytes has been in zebrafish, right? Misha Aron's lab in that Cell paper was really phenomenal paper. So yeah, for the inflammatory component, I haven't looked at it yet, but it's definitely on my radar. Yeah.
Okay, I think so. We move then. Thank you very much. It was again a great talk. Thanks for having accepted. And you are in Canada, so we are in Europe, and also I think it's morning there.
Yeah, it's still morning. Yeah.
So thank you for accepting. And I hope that I write you because I've seen some things that I'm interested. So I will write you myself, and so maybe we can have a collaboration there.
Sounds great.
Thank you again.
Thank you.
So, we move now to Laure, but people can ask if you have a question to ask to Ciaran. You can always at the end do that if there is some time. Yeah. So we. Ciaran stay, I think, with us, and then he can answer the question. Now we move to Laure Bally-Cuif, who is working at Institut Pasteur in Paris. So I think Laure is an old friend. Actually, this was the first idea of this webinar was to because of the Corona. and there is less and less. I mean, there is not many meeting going on, and I thought, okay, having seen the talk of Ciaran, I said, oh, I want also to have my own webinar, and then to do it, I invite some friend and ask them if they want to give talks, and then from that, friends invite other friends and some people. So we came out with a quite good list, and the first one that I thought was Laure, because we have some common projects, some collaboration, and so I very shortly introduce Laure. I think I don't need really to introduce Laure because she's quite well known in the field of neuroscience. So Laure has started in École Normale a few years ago, and from there, I mean, your interest was always, I think, it was always neuroscience. So this didn't really change. She worked first with avian models, and then slowly she moved to fish. And after her thesis, she moved to United States to the lab of Robert Ho. And from there, she did a second postdoc in Germany in, I don't know if you call it the Forschungszentrum, Helmut Research Center in Munich, where she did a postdoc. And very quickly, I think it was in 2009, Laure, is it correct? She started her own lab, then investigating zebrafish early neurogenesis and slowly moving to adult neurogenesis. And we then move also with Laure to adult neurogenesis through several collaboration. And then from Germany, she decided to go back to her home country, to France, where she went to GIF Sorifet, and went on with the same topic, using zebrafish mainly, and working on the different aspects of neurogenesis and I think a little bit regeneration also. And quite recently, I think 4 or 5 years ago, 2016, she moved to Pasteur, and she is now in the Institut Pasteur in Paris. I think I don't need to say more. I'm very happy that Laure, you accepted to give this talk, and I'm very excited to listen to the latest results. Thank you again, Laure.
So thanks, Sepand, first of all, for putting this webinar series together, and thanks for inviting me. I guess it's really nice, and it's important to keep sharing data. So I'm glad you're giving me this opportunity. Okay, so what I'd like to do today is share with you some of the latest data that we got in the lab, and that aim to understand the mechanisms that are important for maintaining neural stem cell populations in the adult vertebrate brain. And so you see here in the background one such population of neural stem cells that I will describe in detail during the talk. But just to start and to phrase the problem about the maintenance of neural stem cell populations. So this is a very long-term process. First of all, in vertebrates in particular, it's a process that can take, or that has to happen during, really months to years. And at the same time, it's not a static process. It's not enough to maintain the stem cells alive, but the stem cells also during all this time have to produce neurons. So it's a long-term and dynamic process of maintaining cell numbers. In addition, it's not just a matter of cell numbers, but it's also a matter of spatial organization of these populations. There's some degree of spatial organizations in the stem cell populations and in the brain, and you can appreciate some of that here in the background. If you look, for example, at the widespread distribution of cell states, which are highlighted by the colors here, the blue and the pink, which I will explain later. But maintaining this spatial organization over time at the same time as cell numbers is important, because it's this spatial organization that will control where the progeny cells are going to be positioned. So in particular, the neurons that will be produced by the stem cells. So it's both a matter of dynamic maintenance of numbers and dynamic maintenance of spatial organization. And at the same time, there's more and more data now that suggests that the maintenance of stem cell populations is controlled both at the single cell and at the population levels. So this means that, in fact, stemness is not so much a property of individual cells, but is more of a systems property. And this suggests the existence of feedbacks between cells in these ensembles that are going to balance the choices that are made by individual cells for their state or for their fate. But at the moment, the molecular nature of these feedback mechanisms and also their spatiotemporal components are not known. And so this is what we are trying to study in the lab at the moment, namely how these different parameters, maintaining numbers, maintaining spatial organization, being dynamic, and coordinating stem cell decisions, how these different parameters are integrated to maintain a stem cell population. And so that's what I will discuss today. So to address these spatiotemporal problem, we use as a model system the adult brain of the zebrafish, and in particular the stem cell population that you see here, and that's located at the surface, at the dorsal surface of the telencephalon. It's the stem cell population of the pallium. So these cells are organized as a monolayer of cells, which you see them here in green, and here you have like a 3D rendering where you see them slightly from below, and here it's slightly from above. So you see this monolayer that's covering the surface of the pallium, and that interfaces the cerebrospinal fluid here and the neurons, the parenchyma, that is generated by the stem cells themselves. The neurons will delaminate into the parenchyma when they are formed. So the stem cells here are, like in mammals, they are astroglial cells. So they, more specifically radial glia, they express astroglial markers like GFAP, S100 beta, or glutamine synthase. And in fact, what you see here in green is a GFAP staining. And they co-express progenitor markers like transcription factors like HER4, which is the ortholog of mammalian HES5, or SOX2, for example. And if you look at this population of stem cells, it shows the hallmarks of very typical stem cell populations. So the first part is that the cells are very tightly juxtaposed. And in fact, what you have here in yellow is a staining for tight junction marker Z01. The cells are also very heterogeneous. You can see that they have very different sizes of their apical domain here, different shapes of these apical domains, a different number of neighbors, and so on. So it's very heterogeneous. You also see very different cell states there, highlighted by the markers, the blue marker, which I will come back to, and one specific heterogeneity in cell state, which is the state in which the activation state of the cell. And here you have activation that is labeled in pink by the proliferation marker PCNA, while many cells, as you see, are negative for this marker and are found in quiescence. So there's a number of heterogeneities in this population that you see here at any given time, but that vary over time and that we have to understand. Now, these radial glial cells, they drive the main neurogenic lineage in the zebrafish pallium, and this slide is the summary of work by many labs. And this is what we understand today of this main neurogenic lineage. So the stem cells are in population here, and they are in equilibrium between quiescence and activation, spending most of their time in quiescence and activating once in a while. So in my drawings, when a cell activates, I'm gonna draw it with a pink nucleus so that you recognize it. Activation means entry into the cell cycle. And every cell, in average, is going to activate once every 1 month or once every 2 months, just to give you a feeling for the very slow dynamic of the process. Once a cell activates, it will then divide, and it can use different division modes, and I'll come back to that. And when the division is neurogenic, it will generate an intermediate progenitor that we call the activated neural progenitor, and that I'm going to draw in orange in the slides, and that itself will divide a few times and generate neurons. So in trying to understand the mechanisms, and in particular the population mechanisms that control the maintenance of these stem cell pools here, we are focusing on two key steps in this lineage that control stem cell maintenance. And these key steps are surrounded here. So the first one is this equilibrium between quiescence and activation, because it's been shown that stemness is dependent on the frequency of activation. Although this is not fully understood, it's shown that the more a cell activates, the sooner it will exhaust. And the second step we are looking at is are the neural stem cell fate choices that are made at division, because it's easy for you to understand that if a cell, for example, decides to divide in a symmetric neurogenic fashion, it will generate two activated neural progenitors and it will get lost. So this directly impacts stem cell maintenance. So today during this talk, I'm going to talk about both, but I'll spend most of my time on this where we have many more data, and I'll just show you some hints of where we stand on the second aspect. So I'll start with this first control step. So quite a few information are known about molecularly what controls this balance at the single cell level. But the question I'd like to ask today is whether and how this control, this process here, this equilibrium, is controlled at the population level, spatio-temporally. And the work I'm going to show you was done in the lab by scientist Nicolas Drey, who directed this work, a PhD student, Laure Mancini, who worked under Nicolas' supervision, and two really fantastic collaborators, Udi Binstock from the Sprinzak lab at the University of Tel Aviv. Udi was involved in the modeling parts that I'm going to show, and Félix Chesson, who is a mathematician, specialist in spatial statistics. So when you look at the position across the stem cell population of the neural stem cell activation events, this is what you see. So what you have here are the two telencephalic hemispheres dissected out of a brain. You're looking at them in whole mount from top, and anterior is to the left. And the stem cells are in green, and so you have this monolayer that I described, and the activation events are in pink. And if you look at a close-up here we have surrounded in white the activated neural stem cells. And you see that these cells are kind of widespread across the stem cell population. So here you have specifically the three cell types that I mentioned in the lineage, the quiescent neural stem cells that are just green, the activated neural stem cells that are green and pink, and they are the ones surrounded in white here, and the activated neural progenitors that are just pink. So the question we've asked is whether this positioning, kind of widespread, was controlled in any way. So is it a stochastic decision made by individual cells, or does it incorporate information from neighbors or information like emergent properties of the population? So to study this, we made two important experimental choices, which I need to describe because they are quite relevant. First of all, we decided to only focus on initial activation events of the cells after the quiescence phase. So the initial recruitment event on a cell of a stem cell. This is because a few stem cells will divide recurrently, generating transiently clustered patterns of cells, of activated cells. But what we were interested in is really the decision to exit quiescence and to get recruited in the lineage. And so this makes this work different from a recently published paper by Prisca Chapouton and Karsten Marr that's focusing specifically on recurrent division events. And the second decision we made is to focus on one territory only, and specifically we've focused on this territory called DM, which is the medial part of the pallium, probably equivalent to the neocortex. And this is because we previously published that the activation frequency was very different in these different neuroanatomical subdivisions of the pallium. The activation frequency and the frequency of generating activated neural progenitors. And if you merge the different territories in spatial statistics, and if you have very different activation frequency, this is kind of blurring the spatial statistics analysis. So we focus on this domain. And what I will show you is a priori valid for this domain only so far. So when studying the localization of these activation events, we in fact followed a very specific and biased hypothesis in the first place, which was that perhaps one factor that would influence the positioning of these activation events, which you see here schematically represented across this stem cell sheet here, is the position of other activation events. And the other activation events are other activated neural stem cells here in pink, or activated neural progenitors, which are here in orange. So basically, we've looked at the position of activated neural stem cells relative to other activated stem cells or relative to activated neural progenitors. And for this, we use several functions of spatial statistics that are gonna plot as a function of the distance here on the x-axis from the cell of interest, the position of the cells that you want to compare with that cell of interest. So for example, cells of the same type here in red across the population. And in red here, you have the experimental curve that's plotting this distribution. And this is compared with what the distribution would be if the positioning was random. And this random distribution is here plotted. It's the dotted black line, and in gray are the confidence intervals. And so if we see a deviation of the experimental curve from a random distribution, a deviation, for example, here towards larger distances, this is indicative of a dispersed pattern of cells and indicative of an inhibitory interaction between the cells that we have considered. And at the opposite, if we see a deviation from random in the other direction, this is indicative of clustering and interpreted as an attraction. And once a deviation is seen from a random curve, then we can analyze what is the range of the interaction that we detect using, for example, rings instead of full circles here that are cumulative functions. This only studies interactions within the considered ring, and we can then also measure, quantitate the strength of the interaction using yet another function that's also taking into account possible differences in cell density in the population that's considered. So using these approaches in spatial statistics, we have first compared the position of activated neural stem cells relative to each other. And what we found is plotted here. This is the red curve, and you see that it's fully random. So we could not detect any interaction between activated stem cells at any given time. However, when we look at whether the position of activated neural stem cells is influenced by the position of activated neural progenitors, then the picture looks different, and we detected an interaction towards an inhibitory interaction, in fact, that we can measure to be at short cell range, so like 1 cell diameter away from the cell of interest. So this is the immediate neighbor of the activated neural progenitor. And when we quantitated the strength of this interaction, we found that in fact, the amount of activated neural stem cells that's found close to activated neural progenitors is about half what we would expect if the distribution was random. So there is a local inhibition of neural stem cell activation in the immediate neighborhood of activated neural progenitors across the sheet. So what could be the mechanism that's controlling this inhibitory interaction? So we had previously shown that activated cells in the stem cell population express delta A, which is a notch ligand. And we have confirmed this here using multiple immunohistochemistries with the delta A GFP transgenic line. And so here you can see, for example, indicated by the orange arrows, the location of activated neural progenitors, and you can see that these cells are positive for delta A. In parallel, we and other labs had shown that one major signaling pathway that's controlling, in fact, promoting quiescence in stem cell populations is notch signaling, specifically notch 3. So the obvious hypothesis is that activated neural progenitors that express delta activate notch signaling in neighboring stem cells to maintain these neighboring cells in quiescence. And so we have tested this directly experimentally by blocking notch signaling in a conditional manner using a pharmacological agent that's blocking the activity of the gamma secretase. And we've used a very mild treatment that is sufficient for blocking notch, but that's not modifying the proportion of the different cell types. So the only thing we are looking at is changes in spatial distribution of these cell types. And what you see is that when we block notch activity, the inhibitory interaction, the dispersion from activated neural progenitors is lost. So directly or indirectly, the effect of activated neural progenitors, their spatial effect, is dependent on notch. Now, what I've shown you here are static analyses in a system that's in fact a dynamic system. So we were also interested in finding out whether the position of activated neural stem cells at any given time could be influenced by the position that activated neural stem cells had before, at earlier time points. So for this, we used intravital imaging. So we had developed some years ago a protocol for this in collaboration with Emmanuel Beaurepaire and Willy Soupato. And so the principle is the following. We are using double mutants for pigmentation genes that are called Casper, that we cross into transgenic backgrounds that highlight stem cells or that highlight specific cell states. So these mutants are transparent at adult stage, and we can use then multicolor, multiphoton imaging to record the stem cells in their endogenous niche in live animals. And so here, this is an example of what we see. So here it's a double detection for fluorescence, which you have in green, and harmonic signals, which appear in cyan. And it's the view you get. It's a live adult fish that is observed under the biphoton microscope. And so in cyan, you see the skin here and the skull that are completely intact. This is a non-invasive procedure. And below, you meet the two pallial hemispheres where you see the stem cell layer, and you see that we have single cell resolution there. You see every individual cell. So because the fish is alive, we can, of course, film the same fish every couple of days. And because the system is very lowly dynamic, we can then make movies and reconstruct the behavior of the cells in situ in the context of their neighboring cells. And we can film the same fish over weeks, 2 months. And so I'm going to show you a movie that was taken by Nicolas. It's a movie of one adult fish called Titi. Titi was 3 months old when it was filmed, and the movie is going to run for 24 days. So Titi is a double mutant, transparent mutant, Casper, and it's a double transgenic also, highlighting stem cells here in green, in false colors, and activation events in pink. And so when I play the movie, you will see, first of all, a close-up coming and with a smaller area, and then you'll see the segmentation, and then indicated by arrows, you'll see some dividing cells that you can follow, and you can look whether the divisions are symmetric or asymmetric, for example. So if I play the movie, this is so the live animal, live brain. And now we come to the close-up. So this is the resolution we have. The little dots are the segmentation. And now we are going to play all the time steps, and you can follow some cells. For example, this one, you see it will divide, making two daughters. One is getting lost and the other one remains. Here you also have two cells. I'm going to play the movie twice because it goes quite fast. Here we are. We can focus on another division, more in the center. Let's see which one. For example, here. And you will see this cell dividing in an asymmetric way. One cell will stay stay green, this one, and the other one will become just pink and get lost in the sheet. Okay, so that's the kind of analysis we can do. And after many years of work, in fact, Nicolas managed to study 3 entire fish with whole mount segmentation and segmentation of the quiescent and activated neural stem cells. And what we get, which is very interesting, is we can record the temporal behavior of each cell, but not only each cell in the context of its neighbors. So we build these division trees that you see here over time. These are the different time steps of the movie, but these trees are resolved in space because we know where they are positioned across the stem cell sheet, and we can look at the behavior of each cell, whether the behavior of each cell depends on the behavior of its neighbors. So we were interested in activation. In fact, what we recorded, what we studied more specifically is an event that is slightly more constrained in time, which is what we call the mother cell event, which is the event that is just preceding division. And so these red dots here plotted on the lineage trees. And the question we've asked is whether the position at one given time of these division events across the stem cell population impacts the position of division events at future time points. And we do that with different time intervals that can be one imaging time point, which is what we call delta T1, or two imaging time points, or three. So we apply the same type of spatial statistics as I showed you before, but over time. So delta T1 is one imaging time point, which is in our case 3 days. So obviously we look at all the delta T1s across all the movie. Delta T2 is 2 imaging time points, 6 days, and so on. And now I'm showing you here the result we obtained with one fish called Mimi. But all the fish show the same trend. And what we observed is a trend for dispersion at delta T4, between mother cells at delta T4. That's what you see here. And this trend becomes significant when the 3 fish that we studied are pooled. So delta T4 is a time delay of 9 to 12 days. So what does it mean? It means that at any given time, the position of neural stem cell division events across the stem cell population is found further away than expected by chance from the position that mother cells division events had 9 to 12 days before. So it's an inhibitory interaction, but with a delay. So this raises a number of questions. Overall, I've shown you that we identified 2 interaction cues that position neural stem cell activation. And these 2 interaction cues are both inhibitory. There's one that's driven by activated neural progenitors and one that's driven by mother cells, and this one is delayed. So the obvious question that comes is whether these 2 interactions are independent from each other or dependent on each other. Then are they the result of active inhibitory mechanisms, or do they emerge from, emergent properties from the population? And what is their relevance, or what is their long-term effect? So these are questions that are virtually impossible to address experimentally. And so we used modeling for this, and this is the collaboration then with the Springsack lab, where we modeled the behavior of neural stem cells in space, over space and time, using a border-free cell lattice. And so to generate this model, we used as input in the model as many experimental parameters as we could quantify from our movies. So in particular, the lineages, the different proportions of cell states and types, the activation rates that we could measure experimentally, the fate choices that we saw occur, and their probability distributions, and so on. So this is the type of information that we enter in the movie. And then we in the model, and then we encoded in the model the spatial inhibition originating from activated neural progenitors. So this inhibition of stem cell activation in the vicinity of an activated neural progenitor. And the question we ask is this sufficient to make the second inhibition from mother cells appear? So here you see some simulations from this lattice with the same color code as before. This one includes the ANP-driven inhibition, and this one doesn't. And when we apply to simulations this time, the same type of spatial statistics as I showed you before, what we saw is that the ANP-driven inhibition is sufficient to make a delayed mother cell inhibition appear with a time interval of delta T4, so 9 to 12 equivalent days in the simulation, while this is not the case when the ANP-driven inhibition is not present. So what it shows is that this ANP-driven inhibition of neural stem cell activation is sufficient to generate a delayed mother cell effect that's really reminiscent of the in vivo, of the effect we see in vivo. So the 2 interactions are likely linked. So what could be the mechanisms that link them? An obvious hypothesis is to think that they might be linked by lineage progression, because the activated neural progenitors are the output of the lineage. They result from the existence of a mother cell. And so to test this, we have modified experimentally in the model the duration of the lineage. And for example, we have modified the half-life of the ANP to increase it or to shorten it. And what we observed when we run simulations this way is that the delta T delay in mother cell inhibition is changed accordingly. So it really responds to the duration of the lineage, suggesting that the ANP and mother cell derived inhibitions are likely linked by lineage progression and by the generation of ANPs. Now, there's been a few studies that were theoretical studies that were conducted on the type of feedbacks that can be exerted to preserve stem cell numbers. Although these models were made without including the spatial component, but just talking about cell numbers, it's possible to apply the feedback on other nodes than the neural stem cell activation rate. For example, it's possible to increase the neural stem cell activation rate, and this will also decrease the frequency of these mother cells close to ANPs. It's possible to change activated neural stem cell division mode and so on. So we've tried to input this in the model, but none of these changes could predict experimental data as well as when we impact the effect of ANPs on the neural stem cell activation rate. So we believe that ANP is really likely feedback on this activation rate. And finally, we used our model to test the long-term or to predict, let's say, the long-term effect that this inhibition could have and these spatial interactions could have by looking at the distribution of neurons. So because there's no migration in this system, neurons, once they are formed, they just delaminate, they don't migrate, and they stack over each other with time. We could use the model to make a 3D rendering of the position of neurons with and without this inhibition. And what we showed is that this inhibition over very long simulations that run over more than a year, this inhibition is important to homogenize the spatiotemporal distribution of neurons. So together, what we showed is that the neural stem cell activation events are not randomly positioned, but they respond to some degree of spatiotemporal coordination. And this is mediated in particular by short-range inhibition from ANPs at any given time. I've shown you that this is directly or indirectly mediated by notch. It's akin to some kind of autoregulatory feedback on neurogenesis. And we believe that this generates a delayed inhibition that spreads or that homogenizes the recruitment of neural stem cells in division across the stem cell sheet. So these interactions generate long-term systems dynamics, and I'll come back to this in my conclusion, and are important for to generate an even distribution of progeny cells, neurons for that matter. Now, to conclude on what I've shown you and where what we think we have understood of how stem cell populations are controlled in a spatiotemporal manner. What I tried to show you is how using intravital imaging of adult stem cell populations in the zebrafish, we can interestingly match the behavior of individual cells and the behavior of the population in situ stem cells in their niche. And I've shown you how we are trying to use this to understand two key steps in neural stem cell maintenance, which are neural stem cell activation and division modes. In the first part, we are more advanced and we've been able to evidence or to provide evidence for the existence of spatiotemporal coordination. In the second case, we are not that far. We are moving there, and I guess that if cell size is really a determinant of division mode, it's fairly easy to imagine how this could be used for spatiotemporal coordination, but we have not shown it. But together we are hoping that this will lead us to understand how local information can be generated in the stem cell population and can be propagated at large scale and long term to sustain homeostasis through life. And I think this is bringing an interesting notion, perhaps, so I wanted to insist on it a little bit, which is the notion of the intrinsic niche, where in fact, as you saw, at least illustrated for activation, it's the neural stem cells themselves and their lineage properties that generate long-term systems dynamics. And this is long-term in the sense that there's no terminal state that is reached in these systems. They permanently auto-stimulate themselves. And our work also allows understanding what are the bases for the fact that we do not reach a terminal state. And these include the fact that the cells involved are transient. These are transient cell types that signal. And so the interactions that we are talking about are transient as well. There's a delay between the input and the output of the interactions that propagate the system in time. And the effects that we are talking about are exerted onto neighbors. So these effects are spatial, and so they propagate the information in space. And interestingly, some stem cell systems in adults are starting to be identified that have similar properties, perhaps, in the mouse epidermis and in the Drosophila gut. And so maybe the notion of an intrinsic niche is something that can be extended to other adult stem cell systems than the brain. So I'll stop here, and I'd like to thank the people who were involved in the work I've shown. So this is my lab, and so the people involved are in orange. And again, I'd like to stress the really enormous contribution of Nicola directing this work and of Laure Mancini, who produced all the data in the second part. And these are our collaborators and funding. So thanks very much for your attention. I'm gonna stop. Works. There we are. Okay.
Thank you very much, Laure. Very nice talk, as expected. If there are any questions. So maybe I ask the first one, so Laure, you correct me if I'm mistaken or misunderstood things. So you show nicely that so basically the cells which get activated influence the cells and which are in the vicinity and would basically control their quiescence, would tell them to stay quiescent. Yeah, this is, and these are the activated stem cells. So have you looked, what about the neurons? Because you show also the neurons, some neurons would delaminate and move a little bit further. Do you have any information? Do you know if the neurons can also influence or they have a role in this?
So the feedback, a feedback from the neurons onto stem cells. This is something that has been shown in particular in the olfactory bulb and so on in the mouse, involving neurotransmitter signaling in particular. So it probably is the case that the neurons play a role. What has not been shown is whether this effect was spatial, So you can control the existence of feedbacks controlling cell numbers is something that is not new. What we show that is new is the existence of feedbacks that have a spatial effect that are important for controlling the distribution of stem cell recruitment. So I don't know about neurons in that particular context. What Nicola and Laure have observed is that in fact when the activated neural progenitor delaminates to form a neuron, this is a process that takes a very long time, really several days. And so it's possible that there is some prolonged interaction between this cell while delaminating and the stem cell. So this is something we have not studied properly, but it may be the case. So maybe not the neuron proper, but at least the delaminating cell.
Okay, thank you. I have many questions, but I mean, we can talk or write to each other. I'll let others to ask. So we have again a question from Emre. I see Emre. I've seen your Emre. So I let Emre ask his question, and then we take the rest of the questions. Please, Emre.
Once again, wonderful talk, Laure. I have a question. I always ask you the same question, I guess. About my question is, do you think these zones that comes from similar progenitors, do they represent similar types of neurons? Like, is there any? Can you speculate? Like, do their architectures look similar? Do they have a similar function, or do they project to similar areas? Can you say a little bit more about, I mean, I know that you're not looking at the function, but I'm curious about your opinion about whether they represent different populations of cells with different properties, or do they have some functional similarities, you think? Thank you.
Yeah, so are you talking about space, or are you talking, about XY, or are you talking about Z?
Both, actually, yes.
So it's a very it's a very complicated question. So there's a number of markers of neuronal identity that will correspond to the arealization, in XY along the planar space that suggests that neurons have different functions. In terms of Z, this is something that we've tried to analyze because we tried for some time to understand whether this was related in any way with the layers that are observed in mammals, for example, and whether the neurons that are formed at different times have different identities, different projections. This is something that is unsolved. We have not been able to find direct relationship with functions that would be layered, So it's possible, but I don't know. We've kind of given up on the subject because for lack of markers. But I agree with you that it's a very important question that is worth studying. Now, in what I showed today, we've really focused on only one neuroanatomical subdivision. For the reasons I explained, and I'm not sure to which extent what we found can be generalized to other regions. And one of the reasons for this is that I think there's a very specific relationship between the internal clock of the stem cells, their clock to activate, and the inhibitory interaction by the activated neural progenitors that makes it possible to see these interactions. And I'm not sure we would see them in other territories. So your question can be extended to basically everything we study. Is it similar in the different neuroanatomical subdivisions? And I don't know.
Thank you so much. I was just going to say, habenula seem to have some interesting features that allows us to relate the function and the birthday. I wonder whether some of these questions could be simply also looked at habenula potentially. Anyway, thank you so much.
Okay, thanks, Emre.
There is many questions. So there is one from, again, Judith. So I'll let Judith ask your own question to Laure.
Hi, Laure. Really nice data, as always. I was really intrigued by this difference in the apical domain that you showed and the correlation to the symmetry of the division. So I was wondering, do you know anything on how this apical domain size difference compares to general size, like cell size differences or morphology differences? Do you know anything about it?
Interesting. Yeah, it's a very interesting question. We don't know. I mean, measuring cell size is something that's very difficult for the radial glia because they have this huge basolateral process that is several hundreds of micrometers in length. So it's very hard to actually measure full cell size. And indeed, initially we did not expect that we would find some very clear correlations just by looking at apical domain size, precisely because 90% of the cell is not apical. 90% of the membrane is not the apical domain, which is very, very tiny compared to the rest of the cell. Nevertheless, this is what we see. We can make the same remark regarding the first part of the talk on the control of stem cell activation, where basically what this also shows is that the interactions that are relevant occur at the apical site. So there's something there, apical, that although very small, it's a key location for interactions. Yeah.
And if I may follow up on that, I mean, I guess it's really difficult to catch with your intravital imaging, but it would be really interesting, right? If you could capture an actual division and look at the asymmetry of the division itself in terms of morphology, right?
Yes. So in fact, in the movies I showed you, so I did not insist on that because we looked at asymmetry in terms of delta expression, but we've also looked. So Laure Mancini also looked at asymmetry in terms of size, and we do not see any difference in size in the two daughters usually, most of the time. So it's not. It's an important question because, in particular, for notch delta signaling, the work of David Sprinzak's lab has shown some years ago that notch delta signaling between sister cells or between cells in general was biased based on cell size, but we do not have any difference in cell size between the daughter cells. So it's something else that we are looking at now, but I cannot. I don't have the answer.
Yeah, really interesting. Thank you.
Yeah, thank you.
Thank you. So I have a question from Olivier Armand, an old friend and a former colleague. So Olivier, can you ask your question?
Thank you, Sepand, for taking my question, and thank you, Laure, for this night talk. It's very nice to talk to old friends. So, Laure, actually, the question I have is perhaps very simple, because you perhaps already said it, but I missed information because your talk is very dense. So sorry if this is a very simple question. In the first part, basically, you see that indeed the activated precursor have a delay action on the activation of the stem cell. And I did not get actually if this is cell autonomous or non-cell autonomous. Is it basically a clock, internal clock of the stem cell that will basically lead to this delayed inhibition? Or is it the different, let's say the activated precursors that will non-cell autonomously go back locally to inhibit further activation of the stem cell? I'm not sure what I said, but
yeah. So in fact, it's because I forgot to mention it. So it's it's okay. No worries. There's two aspects. Okay. But it's not trivial. So we believe that the cells follow an internal clock. And the reason why we believe this is, for example, because when we block notch signaling, if the notch signaling was the only pathway that's controlling whether a cell is quiescent or activated, when blocking notch, all the cells would activate at once. And that's not what is happening. There's a delay sometimes in activation by a few days by individual cells. So this suggests that the cells follow their internal clock, and that according to this internal clock, once in a while, there is a window of susceptibility where they respond to notch. And if you block notch at this moment, then they will activate. Okay. So this is why, in fact, so it's not sufficient to block notch to see all the cells activate. So in the model that we propose, there is an effect triggered by notch via the lineage, generating activated neural progenitors that is superimposed to the internal clock of the cells. And this is what is generating the pattern.
Okay. Thank you very much.
I don't know if that clarifies the...
Yeah, sure. It is.
But I should have been clearer. It's my fault. Yeah.
The next one is Andreas.
Thanks for the talk, and you answered some of my questions already. I was asking about the neurons and typed this when these questions were already asked. But there must be a feedback from the neurons. Also, my question is: you treat the pallium here as one homogeneous surface, but there are different nuclei underneath that do different things. So there must be regional differences in what kind of neurons are produced underneath. Do you have any hint on that? I mean, it goes the same direction as the other questions, but
yeah. So I agree with you. There must be differences. The thing is, we have not been able to identify these differences using markers. I think this would deserve a very systematic approach and a very systematic characterization. But there's clearly, so we are not looking at the whole pallium as one uniform zone. So that's why we precisely focused on the dorsomedial pallium. And indeed, relative to what I showed, like the activation frequency and the possible interactions between cells and perhaps with neurons, these different neuroanatomical domains are very different. So DM has a specific activation rate, but if you take the anterior part of the pallium DA, the activation rate of the stem cells is much higher. If you take the lateral part of the pallium, the activation rate is much lower. And so there's maybe other mechanisms and other input that may be driven by neurons, that are different compared to DM. So it's your hypothesis is very possible. It's just that we have not studied it. So, but it's an important question. I agree. But yeah.
But even within the DM regions, you would have several types of neurons, not just one.
Yes, of course. Yeah. So yes. But when we look at the entire DM, nevertheless, the interactions that we describe are strong enough that they are visible even if we mix, you know, the different areas that you are thinking about. So there might be finer subdivisions and finer interactions that we have missed because we are looking at everything together. But what we see is strong enough to be visible at a large scale.
Okay, thank you.
So we take the two last questions because it's getting late. They're really two last one, and then so I have now a question from Natalie. Natalie, can you ask your question?
Very nice talk, Laure. It's Natalie Juriszek from Trondheim. So yeah, very interesting work. I was curious actually about the interaction between progenitors and the CSF. So I'm particularly interested in CSF and everything that's happening there. And basically what is very interesting about the DM is that they're really in direct contact with the CSF, which is kind of a little bit different from other regions of the pallium. And we also know from the mouse that there's actually like the progenitor cells, they're actually extending out of this pinwheel structure and really sensing what's in the CSF. So my question is, do you have any clue whether there's anything within the CSF that can actually manipulate the behavior of the cells? Because most of what you have described so far is mostly like located within the epithelium, let's say the notch of the neighboring cells or this or that. Do you think there is something there that can actually manipulate the way they are maintained in quiescence, or they're activated, or they're also maybe a spatial organization and so on? I would be curious to hear your opinion about that.
So I mean, most likely there is something in the CSF, and we have not looked at this, we have not studied it, but as you mentioned, and even more than in the mouse, the cells are in, there's no ependymal layer, and so the cells are in direct contact with the CSF. They have a primary cilium that's extending into the CSF, so they must sense whatever is in there. Whether this is important for regionalization is a possibility, because indeed DM is really based in the CSF, but this is not something that we have studied. So we have focused really on these intrinsic niche interactions, but most likely the CSF is playing a role, and whether it does so in the regionalization, it's very possible, but I have no data to answer you. No information.
Thanks a lot. Yeah, thanks a lot. I hope that in the future we'll be able to actually show whether there is an impact or not.
Yeah,
so
exactly.
That's basically the direction that we're taking. Thank you very much.
Rely on you. Yeah.
Thanks.
Okay, so Laure, so Akram is back. Maybe I ask the question of Akram he's wondering what happened to the neural stem cells in neuroinflammatory conditions. So basically, when there is a neuroinflammation, what's happened to the neural stem cells?
But it's a question for you, Sepand.
I know it, but it's your talk.
So neuroinflammation. So several labs have looked at the effect of neuroinflammation on stem cell activation, including Sepand's lab, but also Michael Brandt, Magdalena Goetz, and others. So neuroinflammation is sufficient to reactivate stem cells as well, through a pathway that seems, from the published work, seems to be independent of notch signaling. So there's multiple pathways that control stem cell quiescence and activation that converge, on blocking cells in quiescence or not, or reactivating them when necessary. So we have not looked in our case at the role of, it's been shown, for example, that you can bias cell division mode when manipulating the brain with lesions and so on. So we have, so most likely you will modify the interactions that I've talked about under such conditions as neuroinflammation or lesions and so on. But this is not something we have looked at, but it should be an important next step. That's for sure.
Thank you. So I think we should stop because it's getting late, and some people, because of the corona, maybe they have to go home if they are working because of the couvre-feu. And I want to thank again Ciaran and Laure. Really, really thank you for having give these two nice talk and answering all these questions. I also thanks Cels and Elena who helped us, and Cels for putting us up. For people who can or want, there is another, I mean, it's a long series which will go till the end of the year. And so the next one is on 12th of April. And just watch and check Twitter and so on, all the social media, because there was some complaint from our Asian colleague because it's too late for them, 5 p.m. So the next one, I already asked to Matthias and to the other person who's giving the talk, Diego, that probably we will move we will have that at 2 p.m. or at 1 p.m. I think with that, I can only thanks again people who joined the talk and the two speakers and Cels, and I think we are done.
Yes,
thank you.
So before we go, I would like to thank Sepand for chairing this series and for organizing the whole series, and Laure and Ciaran for your nice presentations. They are actually from my background, from my PhD background, so I really enjoyed them. Thank you very much. And well, I hope to see you soon, and take care. I will close now. Bye-bye.
Thanks very much, everyone.
Thank you.
Thank you.
Bye.
Thank you. Bye-bye.